Okay, we're going to finally talk about filtering, variant filtering here. So we had mentioned this several times. This is going to be a presentation filtering. You'll see a lot of machine learning. If we have time today, I'll have a treat at the end of the day with a new presentation, especially for you. If we don't have time today, maybe I'll try to do it tomorrow. Um, since we don't do chat GPT in this presentation, we'll do something similar if we have time. So we have to see if I can make it. Okay, so in this process now, you saw, we have a whole pipeline, a whole workflow from unmapped reads all the way from the beginning, the top left. We go through, we processed our data, and then we've called variants. We've made a joint call set yesterday. Now we're finally back into the analysis to make sure our call set is good. So we're going to do three things. We're going to filter. I'll talk about that. We'll talk about refining genotypes. And then Aaron will come and talk about evaluating your call set to make sure everything is good. You might think we've done all this work with the haplotype caller. There was a lot of math that went into it. We've been doing this for years. Why do we need to filter any variants? Why are there any mistakes? Okay, and the answer is, again, the sequencing technologies are not perfect, and the human genome is very complicated, and the reads are very short relatively, and so we still make mistakes. Also, the theory behind variant calling is to be very permissive. Get as many possible variants in so we don't miss the important one. We'll filter some out later, but we'd rather it be in the file. We'd rather have some false positives in the file and not miss any real things, rather than be too restrictive and not have and lose some of the real ones. So we're very permissive in variant calling, and then after the fact, we do filtering. And again, we do filtering when we have lots of data. Aaron talked yesterday about why joint calling was important. That's because you can filter. You're more powered for filtering when you see many samples. Okay. We have been doing filtering since like 2011. We have different versions. So we're going to learn a little bit about the versions over time. And we will do some hands-on exercise. We tried to get the latest version ready for the hands-on, but didn't quite make it in time. So you're going to use like the second to latest version of the filtering, but it's okay. There's plenty of time for you to learn everything. We started early on with, and I'll go through all the details, with hard filtering. Hard filtering means you have a very specific cutoff and there's no machine learning involved, and that's that. And that had, its pluses and minuses. It was very easy, but it wasn't as accurate. And then we started introducing statistics and mathematics and machine learning into it. And we have something called VQSR. So you heard yesterday from Emma about BQSR. This is a VQSR. BQSR was base quality score recalibration. This is variant quality score recalibration. And VQSR is built on a Gaussian mixture model, which I'll show you about. And we said that it was fine, but there were issues with it. We started using deep learning, and deep learning we'll go over a little bit today. And that was very powerful and very expensive to run and very complicated. And it only works with one sample. So we said, okay, there's got to be some middle ground here, and that is the latest thing. It's going to be the VETS, with an isolation model will teach you. So all this stuff we're going to go over today, and hopefully I'll give you some details and show you everything. In terms of, there's lots of tools. The GATK isn't the only tool set that gives you filtering approaches. There's other things, different models. People like to use random forests. People like to use SVMs. That's all good as long as as long as you're sure the tool knows what it's doing. There's lots of different approaches. In fact, Nomad, what they often did, I actually don't remember if Emma mentioned this, but Nomad did is they would take the VQSR results from the GATK and they do more filtering on top of that. They'd run a random forest on top of that. So there's lots of filtering, and people try to gear it to their own way. We'll teach you a couple of methods here. Here's the important thing. When the haplotype caller does its variant calling, not only does it output a variant call, but it also outputs some metadata associated with that variant call. And this is, I've taken one VCF record, and instead of making it horizontal across the screen, we've made it a little bit up and down so you can see it better. But really, this is one record. So this is now chromosome 22. position 49,582,364 in the genome. The A means the reference base at that position in the genome is an A, and the variant allele is a G. And that the next number, 198.96, is the quality score. That's the confidence that the site is a variant site. Now, not necessarily that the sample in there is variant, but the site is variant. And the next module, I'll explain to you the difference about the genotypes. This is whether the site is real or not. And so that's quality. So this is a Q score. So remember, Q90 is 99.9999999% confident. This is even more confident than that. So this is probably a real site. The next field is one big text string, but we've broken up into the lines. It's one string divided by semicolons. And each one of those things is metadata. And some of them are very important for the filtering. Some of them are not. But I'll point out a couple of them. The DP means depth. That means at that position in the genome, there are 87 reads. Well, now you know why we're so confident in the site. There's 87 reads, not 30. There's 87 reads. That's a lot. MQ is the average mapping quality. Now, the mapping quality, I don't know if we mentioned this with alignment yesterday, is the confidence that each read was aligned well. And the maximum usually is Q60. BWA doesn't create anything larger than Q60. So the average mapping quality here was over Q51. That's very high. So that's a good site. The QD is another important one. That's the quality of the site. So 198 divided by the depth. As the depth goes up, the confidence in the site should go up, right? If the depth goes up and the confidence does not go up, that's usually indicative of something bad. So we use all these. These are examples of things we use. SB was strand bias. Aaron mentioned strand bias yesterday, where you should have reads overlapping the variant from both directions. Okay, so all of these things go into models, and this is the thing that feeds the training and the evaluation of filtering. So imagine now I've taken any two of those features, any two of the metadata annotations. We call them annotations. And I plot one on the y-axis, one on the x-axis. The details, which ones are important, but imagine this is what the spread looks like. And I want to say some are true positives and some are false positives. The things that have high qual by depth are good, and the ones with low qual by depth are bad. And the ones that with lots of strand bias are bad, and with low strand bias is good. Okay, and so we can plot them there. And then in the olden days, we chose hard cutoffs. We said anything to the left of this line is bad, and to the right of this line is good. Now, it worked fine for 99% of the variants, okay? But it's a very, as we say, a blunt-edged instrument, that blunt-edged sword. So it means, let's say you have a good site that's just barely bad. Everything's a distribution in terms of a bell curve, and it's just on the edge, just barely over for one of the dimensions. But the other one is perfect, or all. In 10 dimensions, it's amazing, but in one dimension, it's just slightly bad. We've just lost that site because it failed the filter. And let's say you have a bad site that's just barely passing in every single one. Well, we just passed our filters, but it's not a great site. So we very quickly realized we should start doing some machine learning and some statistics. And the first thing we did was write something called, again, variant quality score recalibration. The nice thing there is you don't have any hard cutoffs. All you do is you take these features, you learn a model, and you create a score for each variant. And we'll teach you how we do that, but you score them. Then you can say, I'll choose a new cutoff. I want to make sure I have no false positives. So I'll choose only the things that score at 90 or above. Or I want to make sure I don't miss that one variant that's causing the disease. I'll take the things that are more flexible and more liberal. I'll take things that score 80 and above, for example. And you can do that. You can set your own threshold with these models now. And so all the statistical models that you're going to learn about all produce a score and allow you to choose the cutoffs. And there are always recommendations, but ultimately it's up to you to choose what you want for your own projects. Okay, for in the rare variant side, where they don't want to miss the one mutation that could be causing disease, their cutoffs are very low. Okay? In population genetics, they don't mind missing some SNPs here or there. Okay, so this is the hot concept. I just want to, you're supposed to imagine this as like IgV. This is this span across the genome here. So those green bars on the bottom show confident regions in the genome that we know we trust the variant calls. So imagine you have a validated VCF. You have a truth set. So we had NA128778. Aaron mentioned she's been the most sequenced sample in the world. Actually, I want to show you a new truth set that we're starting to use instead. But imagine you have some sort of truth data, which you need for this. You go into your own, so the unvalidated VCF is your call set, the one you made. And you go there and say, look, I can see from my validated VCF that there are, imagine these stars are all variants, okay? That they overlap the green and green. So that means there's a variant present in my data set and a variant present in the truth data set, and they match. So now I know that that one in my own data set is real, and I can use that to train on. Okay? Now, I don't want to train on your data set because your data set was sequenced differently, had different samples, different sequencing machines, different dates, different everything. So I want to train on my own data set, but I only want to train on the real sites. And I also have sites that I know are not real because these red stars represent variants that I can see in my data set that I know are not real because they're not in the. So for example, what we do at the Broad is whenever we create a call set, we add in NA1287A. Even if she's not part of the project, we always add her in. We sequence her all the time. We must have sequenced her thousands and thousands of times at the Broad. Okay, and this way, when they get new machines or they update the chemistry, we always have new models with her. Okay, so she's always included in the call set. Actually, we include her husband and daughter also. Okay, so you have a trio. So this way, you know you have some truth data to train on. We know all of her real variants are real, and when things that are called in NA1287A that aren't really there because we know the truth, we know are false positives. So we can train false positives and true positives. That makes sense? Very important. Okay. So very common truth sets are something called Genomes in a Bottle, NIST. NIST is the National Institute for Standards and Technology, I believe. It's a government, US government agency that is trying to make standards. And they have created a truth set and have sequenced a whole bunch of, including NA1287A, her entire family. She was a great grandmother. She's now dead. And they sequenced her children, her grandchildren, and some of her great grandchildren too. So they have a whole family now that's sequenced at very large depth. So it's a pretty strong truth data set because you can see which mutations are inherited now, and you know which ones are real now. Okay, I'm going to teach you the one on the bottom right. Okay, the SynDip, we call it synthetic diploid. SynDip is synthetic, a fake diploid. I'll teach you about that one. So this is interesting. Okay, I cannot pronounce this word. Hidatiform. Okay, so what that is, I'll explain to you biologically what's happening. Sometimes in biology, you have a sperm and egg come together, and turns out the egg didn't have any DNA material in it. So obviously it does not create a fetus that is viable, but you have a sperm and egg that come. The sperm has DNA from the father, and the egg does not have DNA from the mother. And it becomes, this becomes a thing, and usually it miscarries. But if you can grab it before it miscarries, you now have something very interesting. You now have single DNA molecule, single DNA embryo. Okay, and so you can't make it a human, but you can sequence it. All right. Now, actually, it's truly diploid. There's two copies of the sperm in there, but it's still the same sperm. So it's diploid, but really this basically haploid. Really one, Okay, so that doesn't mean it's easy to do variant calling, but it's a lot easier to do variant calling on a haploid genome than on a diploid one, because we can check to see anytime we see a heterozygous SNP call, we know it's not real. We know there's some error there. So you can throw all those out. And that's why you have those confident regions. We say these are regions where everything is always homozygous, reference or variant. And anytime we see a lot of heterozygosity, we say, let's ignore this region. It's a bad region in the human genome. So we did this for two different ones, CHM1 and CHM13. So we have two copies now separately, and we can create the truth data sets for each one separately. Then we can take them both and throw them into a dish and create a synthetic individual that's half, we'll say one is the father and one is the mother now. So it's a combination of CHM1 and CHM13. So it's a synthetic and artificial diploid sample. But we have a much stronger understanding of what the truth is now for this individual. And we've thrown away NA12878, and now we use this sample instead. And this has become one of the standards for truth. Okay, does this make sense? So if you wanted to, you can grab one of these. You can use NA12878. You can use your own truth sample, and always include that for evaluating your and training your models. So this is back into the olden days of VQSR. And this is what we did early on for the 1,000 Genomes Project. What you're seeing on the left is we take our VCF, the joint call set that you have created. This is your own call set, your own samples, and say, look, I'm only going to evaluate sites that I know are real, are good variants. And so that's what we've done here. The red, anytime, so we're plotting two different of those annotations, of those features. So the one on the bottom is that quality by depth, the QD, and the one on the left is a strand bias. And we're only kind of showing you dots of where a variant in your in our call set here, this was actually N812878, showed up in HapMap. HapMap, again, is a big public data set and very strong reliability, and those are red. And also, if it showed up in something called dbSNP. dbSNP was also a catalog of variants of SNPs and indels in the US, supported by the National Institute of Health, that is less reliable because it wasn't the government creating call sets. It was PIs and researchers contributing their variant calls. And this was in the early days when they didn't use the GATK. We used the GATK, but they didn't. And so I know a lot of those things are errors. So if you see those yellow ones, so there are yellow under the red too, but there's a cloud up on the top left that are those likely dbSNP errors. I know what those are because I told them they were errors. These were indels that were miscalled into SNPs because they didn't use the haplotype caller. So this is when we started making everyone change, when we started showing that plot. Okay. But notice that the heterozygous and the homozygous SNPs and variants, they kind of separate into two different clouds because they have different features. And you can very easily see that when you model it like that, when you train a model. Now you've trained a model on the left, and you take it, you go back to your call set and say, look, I only looked at sites that I knew were true. Now let's look at every other site in my call set. Every other site in NA12878. And let's evaluate, see where they fall. Let's plot them here. And notice there are, again, 2 or 3 clouds. There's a whole bunch of purple. These are things that we filter out because they're not anything like, they're more like those dbSNP errors, right? These are a lot of errors. We see a lot of them that need to get filtered out. There's a whole bunch of what clearly look like to be heterozygous SNPs. And then there are very few things that fall on the homozygous side. So just a test here. Here's a trivia question. Why are there very few variants that show up in the novel variants that are homozygous variant? Only people in this room can answer, unfortunately. Sorry to those in the other room. Why are there no or very, very few homozygous variant SNPs here? Anyone want to give you extra lunch if you can get it right? No guesses? All right, I'm going to give you a big hint. Hardy-Weinberg is the hint here. So what is the Hardy-Weinberg theorem? What does it say? That if something is homozygous, it's going to be high frequency. All right, so a homozygous variant will be at high frequency based on this is genetics. If it's high frequency, it's almost certainly going to be already seen in HapMap or dbSNP. So we would have seen it already. There are very few SNPs we'll see that'll be homozygous that will actually not have already been seen in someone else. And this is meeting our expectations. Okay, that was a side point. But the important point here is that we need to, set those thresholds between what are clearly bad and clearly good, and they kind of bleed in. Sometimes it's hard to see that dividing line. Okay. VQSR was great. The problem is that it handled large call sets. there are a couple of problems. One is it used a Gaussian distribution model, and they did that. We did that because it made sense at the time in terms of multidimensionality of your modeling. We would model 12 dimensions. Okay, we only plotted 2 here because I can't draw in 12 dimensions for you on the screen, but you were modeling pretty complex things. But most of the data doesn't really fit a Gaussian model, so it wasn't great. Also, it was as the call sets got bigger and bigger, just like joint calling got harder and harder, the variant filtering got harder and harder. And it doesn't really scale well, ultimately. So it scales to call sets, just like Aaron was mentioning, to 10,000, 15, 20,000, but beyond that, it's hard. Okay. And the other thing is we don't always have great negative training data. So there's truth data, but you haven't always sequenced the CHM hydatidiform mole or NA12878. So it's not always easy to get the false positives in your data set that are false positives. So we said, let's try something new. All right, let's also take a look. We can do something better with deep learning here. And we can also keep into account structure of the genome. So with this is a very complicated plot to understand. Let me just explain it to you. For any one of the colors, that represents a SNP, a type of transformation. So just for example, the purple on the bottom right is an A to T transition. An A turns into a T. And you can view this as actually mistakes, how often there are mistakes in the sequencing, for example. Now, within each color, that bottom right key tells you what the bases around that are. So there's a big purple tower, right, on the left. That means that when an A turns into a T, often there's a T on one side and T on the other. And when that's an error. So when you see a T and then a T and a T, usually the real thing is an A, okay, more often than not. So this is an error plot often. And so we know that the TAT turns into TTT in Illumina machines more often than it should. Well, that's very useful information. Okay, to know that these sequencing machines make mistakes. That's very useful. We didn't use that in VQSR. We should be able to use that. Okay? Does that make sense? I don't need you to understand that entire plot, but just the concept there. All right. The next few slides, I just want to get my laptop. The next few slides will be a little complicated. I don't want you to go into, I didn't want to scare you with the math of the half-life caller. We're now going to talk about some deep learning approaches. I want to keep this very high level. If you are a deep learning expert, this is not going to be for you. Okay. If you're interested in how the approach is, I'm going to keep it again high level. I'm happy to go into more details later, or even point you to a paper you could read offline. Okay. So tensors are the entity, the data structure used for deep learning. So what we do actually is we're going to represent now we're going to represent the genome. We're going to traverse the genome and model it and try to find out where errors occur. Okay, what are errorful sites and what types of structure leads to SNPs? And we can do this much more powerfully with deep learning. So we have an example sequence here, TA, TAA, TG. This is just an example, a toy example for our. So we turn that into what's called your tensor, this is like a tensor encoding thing. So a column here is one channel, okay? And notice though that you can encode. So on the first column, you have a 1 on the bottom. That's your T channel, okay? That makes sense. And you can encode ends also. So you code ends are I don't know the answer. So there's a lot of inconsistency you can encode and unknowingness you can encode in this. Again, I have a toy example. So if you know about multiple sequence alignments, this thing on the left should look familiar to you. This is the way of expressing, hey, look, I know that certain types of motifs, certain types of strings of letters are conserved in the genome, or some create more errors. And that plot on the bottom shows you, if you look at all these things together, you see that the G and T in the middle are more conserved than the other things. But if you were to convert that into a single, bit set of zeros and ones, like on the top right, a consensus, if I say, I'll just take within that that column the most common letter at that position, you lose all the information. But if we take that position, the weight matrix, which we had in the previous slide. So here, if we have zeros and ones in the previous slide. If we turn them not to zeros and ones, but to actual floating point numbers, probabilities or likelihoods, we can actually encode a lot more information. And so it's not just an A turns into a T all the time, but we encode this. So that makes sense. What we're trying to do here is try to find patterns in the genome, and not hard code it to one transition. Right? So this is the position weight matrix, and you can do that many, many times. Do that over and over again. And so we do something called convolution. Convolution is a mathematical term, which means a blend. A convolution is a blending of different functions. So we're basically taking those motifs. And again, this is very high level. This is fun deep learning. And you blend these things across many, many, many thousands of motifs. You're trying to have the computer learn a model of what happens in the genome. Don't worry if you don't understand it. Don't worry. Okay, I can see people saying, I don't get it. Right? We're going to get to the fun part soon. So we're going to have it learn all about the sequence and what the changes look like. So here's some examples. So it learns some stuff. It definitely learns. Okay. So usually letters on top mean it learns that those are true alterations in the genome. And things on the bottom are, oh, I see false positives. So for example, let's look at the bottom left one, the Cs. What it means is when there's a bunch of Cs in a row in the genome, the sequencers tend to make mistakes and make them more Cs. That's what that's learning there. So this is a homopolymer run, it's called. Lots of letters of one type start to bleed. So I had mentioned this before that with Illumina sequencing in particular, if it sees the same letter over and over again, once that letter stops, the machine doesn't know, and it keeps hallucinating that same letter again. So that's learning on the bottom there. And you can learn lots and lots and lots of different motifs. Okay? And all you're doing is, all you've given it was the reference sequence and your VCF. Okay? So that's great. That's wonderful. And that's called, we have a one-dimensional model that does that. But we also have a pretty cool thing called a two-dimensional model. Let's say we want to give it also the reads and learn from there too. Now, this thing on the right, we'll talk about, is actually not 2D. This is a three-dimensional structure that I can't even, that I cannot display for you otherwise. I think I have a picture of it when I try to show you a 3D. But what you're now is, you're not just representing the reference and the read, you're also representing what's happening in the reads. And you're throwing this all into one big model and trying to find differences from the reads and the reference all together and let the machine learn the differences there. Okay, and all the features that go into those reads are in there too. So that's what I mentioned. Okay, so here on the bottom, you can kind of see there's a three-dimensionality to it. And this is actually what the machine is learning. And what we found here, I know this looks pretty clear, right? You can see the SNP. Everyone can see the SNP? Yeah? I hope no one's saying yes, because it doesn't make it. Okay. So this is actually a three-dimensional channel that you're seeing here of a SNP that it has learned inside the data. So what am I looking at here? So it's a G to A. So G in IGV, we've used the same colors as IGV. G is brown and A is green. So look over here, right in the middle. I don't know if you can see them online. Right at this position here, there's brown and A. And you see a SNP right here. It has learned from the data, from the features of it, that there is a SNP in this position in the genome based on the reads. Okay? And it can learn other things too. It can learn deletions. So insertions. So an insertion of a T, for example, this pink thing here means there's nothing in the reference genome, and there's a red, T is red in IGV, there's a red line here. It is learned within the whole sequence right here. This is all, this is all the reference. And at this position here, where there's nothing, there's a T. So these are all channels. This is all, a 3D deep learning channel, tensor, that you can see actually insertions. And you get similar things over here with deletions. There's nothing in your sample where there are stuff in the reference. Okay? So I hope that was crystal clear. Everyone understood that. But again, this is the kind of fun thing we've done with so this is a convolutional neural network. CNN, and there's a caller that puts it all together called CNN score variance, and it's pretty good, pretty powerful. If you take a look at its, the best in class results, it really outperforms everything else. It's much better than VQSR. Okay, so if you have a single individual sample, you want the best quality results. You have a clinical sample, you'd run something like CNN score variance. And we're not going to go through it now. I think the hands-on we'll do later will not have CNN. Will it have CNN? It has the 1D model of CNN, okay, because the 2D model is slow and expensive, which I'll talk about. But we have the command lines here if and when you want to run them. Okay, and then again, the first two commands are either 1D or 2D models. 2D is with the reads, 1D is without the reads. The first step is to train a model. And then, remember I mentioned, the training really just creates a score. It scores every variant. And then you decide where to cut off your variance. Okay, and that's what that second step is here. These slides, again, are all available to you, so I'm not going to go through all the details now, just because I see the interest of time, I don't want to get too late. Okay, so CNN score variance is pretty awesome. It does really, really well. Okay, and it does not matter so it can learn it's a new model for every data set. Doesn't matter, it doesn't rely on Gaussians or anything like that. Great. Here's the problem. It only works for a single sample. So when you have a joint call set, it's not going to work. And most people these days, we just got through telling you how important it was to have a joint call set. And also, it's really slow. Deep learning is not a fast process for training. So there's inference, okay, and there's training and all this stuff. So applying it is pretty quickly, but training the model is not. So we didn't stop there. We decided to do something in the middle. And there's a new filtering tool that we use now called VETS. Now, stands for Variant Extract, Train, and Score. Extract features, and you'll see there's 3 steps to it. The extraction step, the training step, and the scoring step. Okay, so we decided to kind of rework the VQSR and do something in the middle. So it has to be fast, has to scale, it has to perform well, and do everything. It takes just the pros of the CNN and the pros of VQSR and try to not do the cons. And so I think we've gotten pretty close to that with VETS. Okay, when Aaron talked about his massive call sets of 300,000 samples, I think it was, they used VETS. They did not use VQSR. They did not use CNN. They used VETS, and it worked amazingly well. Okay, so this thing can handle hundreds of thousands of samples, which is pretty amazing. Okay, and it's fast, it's scalable, it's quick, everything is great, and it does not cost a lot of money. When Aaron talked about the prices, the cost yesterday for call set creation, I believe that included the filtering too. So this is just a plot showing that it outperforms VQSR. In the interest of time, I want to move a little bit and kind of show you how to run it. So there are 3 steps. In the future, when we come back here next year, the filtering hands-on, we'll use VETS instead of VQSR. Today, we're going to use VQSR. We didn't have time just to finish this one to get this over the line to you. But next time, we'll be using VETS. And there are, 3 steps to extract the annotations out. So remember, the annotations there these are the features, the metadata associated with all the variants. You pull them out. Okay, the -O is your output, and it's just a header prefix. You train your model, and the training of the model is pretty quick. It runs pretty quickly. And the input there to this training the model is the output from the previous step. And then you score things. Okay, you score your variance. Pretty simple. Extract, train a model, score things. And then you're ready to filter all over again. It's a very, very quick process. We break it into 3 steps. So along the way, we can make sure things are working well. If you want to parallelize things, you can do that. If you want to reserve, the training step is obviously more computationally expensive than the other ones. So if you want to reserve a different virtual machine with a lot more memory, for example, you don't have to do it for all 3 steps. So we break them up into 3 steps, and you can just pay for what you use in the expensive step. That's why we break it up. Okay, so that was filtering at a high level. as I mentioned, after lunch, Anton will lead us through a hands-on exercise for filtering. I now have genotype refinement, but let me just stop and take questions for those who have it. Again, anyone next door is welcome to come over with questions. So questions now on filtering before we go to genotype refinement. Yeah, here.
So if I get it right, the 2D model should be more accurate than 1D model, right? But I saw the publication has the different conclusion.
Wait, the 2D model, the 2D model is definitely more accurate than the 1D model. Oh, less accurate. It wouldn't be really? Okay. Oh, so it could be in maybe one sample, but it's definitely not in. Oh, okay. Show me what we can look at it afterwards. I think like the 2D model is definitely more accurate, and you can learn a lot more features from it. But it also is really slow, like really, really painfully slow. So we don't really use it much. Yeah. Good. Happily. Yeah. Okay. Good. Other questions? Okay. All right. Let's move over to genotype refinement. And Aaron talked about this a little bit yesterday in his hands-on and mentioned an example of why you'd need to do this. His example was the mother was homozygous reference and the father was homozygous variant. And so since they're both passing on an allele to their progeny, to their child, the child should be heterozygous. The child was not heterozygous in multiple cases. That makes no sense biologically. Actually, there is a sense kind of where it could make sense biologically, which is tricky. And that is when you got the genotype wrong in the parents. And that, for example, let's say the mother was not actually homozygous reference, but the mother had a deletion, a large, large deletion. And so actually she was heterozygous, reference, and a deletion. And what she passed on was the deletion to the child, and then the child could have gotten the SNP from the father. Now the child has a deletion in the SNP, and it looks homozygous. So that's a rare case. For the most part, the problem is with the likelihoods and not with this case. But we saw this case enough. Obviously, it causes a Mendelian violation. So that's how we know that it's the first thing we look at for large deletions. But for the most part, when you see a Mendelian violation, it's an issue with the, not variant calling, but with the technologies. I'll show you some examples of where things are very strange inside the reads, the sequencing data itself. Okay. Now, I'll say again, when we run our production pipelines at the Broad, we don't run genotype refinement because the researchers don't want us to. And that's because we don't have access to the metadata. So we don't know if samples are male or female. We don't know what ethnicity they're from. We didn't know if they're families. If they were to tell us that, we could easily run this stuff and make things better. But we aren't given that information in our sequencing lab. If you do have that information, this makes your data much, much better. So we require the researchers to unfortunately run this themselves. Okay? I don't know why we don't get the data, but that's just what it is. Okay, so why are genotypes so important? Why do we need to know? I actually mentioned a case yesterday of the cis and trans, knowing when two genotypes are either on the same haplotype or not. Also knowing if you have recessive or dominant genes, if you have mutations there, that's a big issue. Knowing exactly what the genotype is crucial for understanding the risk of disease for any one of very deleterious alleles. Super important to do this. Okay, so in IGV, I just want to make sure we understand the difference between, I had mentioned this before, a variant call and a genotype call. The variant call is the site in the genome, is the site a variant site. And the genotype is it heterozygous, homozygous, okay? And there's two different confidences. Your qual is the confidence that the site is truly variant, is biallelic. It's not monolithic, okay? The GQ, the genotype quality, is the sample's genotype correct? We talked about the math for that in the haplotype caller. So here in this example, notice that on the left, Okay, I want to jump in here. Sorry. Okay. So on the left, you can see that the reference position here is a C, and you see the T SNP here. Okay, now look at, first of all, this shows you the balance here. There are 39 reads, and almost all of them are T's, but there's some C's. Now, this should be 50-50 if it's a heterozygous SNP. This is why the genotyping quality sometimes is inaccurate. It's not that the haplotype color did anything wrong. It did it the right thing. It sees some C's here. So, well, is that heterozygous? If it's at a 90, so out of 40 reads, if 4 of them are C's and 36 are T's, what is that? Is it heterozygous or homozygous? It should be homozygous, right? Mathematically. But why are there C's? If it's homozygous, why do I see any T's? Okay, somatic mutation is a good answer. That's a fine. It could be contamination from another sample. Sure. I mean, those are terrible things, but those are possible. It also, there's many other reasons. The sequencing is imperfect. The alignment is not perfect. Could be, again, bacterial contamination also. So there's many reasons you'd see something like this. It's not the variant caller's fault. It's doing the right thing. It sees it right there. Okay, but we still have high confidence that the site is real. We might have no confidence in the genotype. But we have a very high confidence in the site, and that's important. Okay, so I don't want to filter this out just because I don't have confidence in the genotype. Because I know this thing is real. I just don't know exactly what the genotype is. Good? Okay. We mentioned a little bit about this now. So what we use is so we use to improve these calls are 2 major things. If you have family information, if you know that this is the child and you have the 2 parents, we can learn a lot from that. Again, there are de novo mutations, expected Mendelian violations, but very few of them. So if we see Mendelian violations, we can factor that in. Also, if we know the allele frequency in the population, so Emma talked about nomad, we can look into nomad. If you know you have an African sample, well, I can look at the frequency of this mutation in Africa as a nomad and apply that as a prior probability. And so we can use it. There's a lot of information we can use to power this. So I talked about Bayes' law yesterday. Here's this is going to be important to understand the difference here. So their posterior probability, or probability of something being true, is equal to your prior probability times the likelihood. Now, the likelihoods we learned about yesterday, we got those from the haplotype caller. So those are going to come into us for free. Now, the prior probability is how much we know from the chance of that happening based on, again, an African sample, the frequency in the population. So I know this variant happens at 90% of samples of African ancestry, and this sample is of African ancestry. Well, my prior probability is pretty high that this is a real variant, right? Okay. So they're for the high level, the chance here. So we have a toy example of if you see someone walking in with an umbrella, which is very practical today. What is the probability that it's raining outside? So it wasn't raining before. Is it raining now if someone walks in with an umbrella? So that's given there. The posterior probability of it raining, given that I observed an umbrella, is the likelihood. So that it's when it rains, I see people with an umbrella, and the prior probability of whether it has rained or not. Okay. So there are a couple things. This is the flow for improving and refining genotypes. And I'll go through this step in examples, okay? But you calculate the posteriors, which means you apply priors. We have the likelihoods. We apply priors. We do some filtering and then some annotation. And Emma talked about annotation, so we won't do too much about that today. Okay, so that is the command line to use. It's pretty simple. You call the tool, calculate genotype posteriors. You give it A-R is the reference. -V is the input VCF. Now the next 2 are optional. -PED. The PED is a format for showing pedigree, for storing pedigrees. So this is a family pedigree map, and there's a standard format for that. And then a VCF shows you, it reads frequencies. So you could use nomad.VCF, for example, here. And it'll learn the frequencies from that VCF and apply it in. And then you have your output. So let's look at an example here. All right, so notice here that the original call is homozygous reference. Okay, so let's jump down to the bottom panel here. And you see an IGV. It's about 80, 85% T and 15% not T. Right, so this is, we expect it to be 50-50%, or at 85-15. Could be a lot of contamination, could be something somatic. It's probably not somatic, because the variant, or if it's a somatic mutation, it means you have the mutation, and then you cancer turn it back to normal, right? So probably not a somatic mutation. Okay, in this case. So I can see the reference allele is green. Green is an A. So that means I have high confidence in the site, but my likelihood is low. So if you look at the likelihood, you'll see three numbers. The first one is homozygous reference. The second is whether it's heterozygous, and the one on the right is homozygous variant. Number 0, the lower for likelihoods, the lower the better, because we're Fred scaled. So this means that we're going to make a call that it's homozygous variant, but it's the difference between the best and the second best is only 3. So I don't even have 90% confidence. I have like very little confidence in this genotype, which makes sense. Look at the allele balance. It's all weird. Good? Okay. So let's say, though, I have now priors. Let's say I know that the red is the prior. Let's say I know that this variant is a nomad, but it's a nomad at a very low frequency. Very few people have this variant in nomad. So the chance of my having here is low. So I apply that prior, 2 over 1,000. That's how many people have it in nomad. And I apply that as a prior, and it now shifts my posterior probabilities. So I have a much better sense of what this real genotype is. It's almost certainly heterozygous. And I don't have 99.9999% confidence, but I do have almost Q30, 99.9% confidence, because I can see it very rare and the chance of my having it here. So priors don't completely overtake the likelihoods, but they do affect it. That makes sense? This is very important. Very important to get your genotypes right. Okay, let me give you another example. Same thing here. Look at the data. Okay, the likelihoods. You can't even see them here. So the likelihoods show 0 and 0. So you're just as equally likely to be heterozygous as homozygous variant. I have no idea. No idea. Okay, so my genotype quality is 0. My confidence in the site, my variant quality is sky high, through the roof. I know there's something here, but I have no idea what the genotype is. All right, now look at my priors. This is a very common variant. Very, very common in nomad. Well, so if it's very common, we talked about Hardy-Weinberg, most likely this is going to be homozygous variant. And those prior probabilities move the scale over. And again, we're not Q100, we're Q16, which is a lot better than Q0. So that's what these do. That's what the priors are doing. And what this plot is showing basically is as we fix the posterior probabilities, as we fix these things in the genotypes, we're getting much more accurate. You're trying to get to the unit line in here, and this plot is just showing a much larger accuracy in truth data. So again, we can do this with NA12878, or with the CHMs, okay, those moles, and we can see that our genotypes definitely get improved. And on average, you have a Q10 increase. Okay, that's pretty high. It's a significant increase. That's an order of magnitude. That means 1 order of magnitude increase. 10 times more confident now. That's very good. Very, very important. And it doesn't affect the incorrect calls. Those stay about the same, which is equally important. So the correct ones are now 10 times better, and the incorrect ones are just as equally bad. Okay, and the other thing we talked about is if you know the parents. So I'm not going to go through all the details here, but again, if you know your parents are both homozygous with different alleles, the child should be heterozygous. And you can apply your probabilities or your prior probabilities of being a Mendelian violation. Now listen, it's very important to say we have to allow Mendelian violations. We know there are de novo mutations. So the priors can't be impossible to allow de novo mutations, but they have to be the right. So again, if you're assuming there are about 20 de novo mutations per genome, then you can just put that into the mathematics. And you also have to account for the fact that let's say both parents are homozygous reference and the child is homozygous variant. Is it possible? I mean, it's possible. Is it likely? No. The possibility is the chance of having Mendelian violation squared, the same Mendelian violation in both haplotypes. But it's possible, so we have to account for it in the math. And so that's all accounted for, and that's what we've done here. And I'm not going to go through the math and scare you after I did that whole deep learning thing with convolutions. Okay. And so once you model it there, it's very, very simple to improve your genotypes based on, again, this is based on family information on the pedigree, and it's all modeled in terms of the probability of having Mendelian violation through. That parameter is settable. So if you know you're dealing with an organism that's not human, and the Mendelian violation rate is expected to be higher, obviously you can set that rate. But for the most part, you just want to use, if you're dealing with human data, we know it's pretty standard with that rate. And this is, again, how you would run it. You've taken your genotypes, you've gotten down, and then we actually want to filter them too. So just like you filter a position, you can also filter your genotypes. So again, if you're looking at a rare disease study, you only want to look at genotypes that are confident. So we had talked about this yesterday. I mentioned that Q20, Q30 is about your cutoff. That's the example we have here. What you can do is you can filter out a genotype. So imagine you have 10 samples in your VCF. I don't have to filter out the whole position. I can say, look, 9 of those samples are good. One of them, the data wasn't great, and I want to filter that one sample out. And that's what the variant filtration allows you to do. So this is not the same thing as VQSR. You're not filtering out the site, you're filtering out the genotype based on your genotype quality. Okay? You don't have to do this, but this is easy. If you do this, it makes your analyses downstream a lot easier because you know to ignore these low quality genotypes. And then finally, you can run annotation. There's lots of tools to do variant annotation. The one I'm showing you here is one built into the GATK. It doesn't do biological annotation that Emma was talking about in terms of deleterious or anything to do with frame shifts. This is just to note that you might find a site, a position where you see a Mendelian violation, what looks to be a Mendelian violation, and you can put that up in the info field by running this. And this is something that you can hand off to researchers and say downstream, go validate. I have 30 of these inside my BAM file now, my VCF. Please go ahead and, grep for a possible de novo, as the GATK can determine that, and go look and revalidate or reconfirm these 30 sites. And so it's very easy to annotate your VCF with the fact that something is potentially a de novo variant, which is important. Okay, so I talked about this a little bit, so we'll skip this piece. Okay, so just to kind of give you a sense, we took a real, I didn't want to go, it's a real clinical case here, so I'm not going to give you all the details of which sample it is, but I'm trying to give you a sense of the order of magnitude of how many de novo mutations you see in a call set. So again, this is a call set of 3 samples, mother, father, and child, real clinical case. It's not any 12878, I promise. When you first made your call set, you have about 3 million, 4 million SNPs and indels in there, and about 417 of them were pretty high confident, what looked like to be de novo mutations. Now again, we're expecting 20, 25, maybe 30. So we refined the genotypes, like I showed you, and we're down to 17. That's very good. Okay. When you go down to the filtering of the genotype quality, and for de novos, often you might even want to get a Q60. You only have 8, which means it's a lot easier for researchers to go in and manually look at these things, or to go back and revalidate them inside a lab. You're not looking at hundreds of sites anymore. This is why we do that. This is again back to what Emma was showing with Nomad. In terms of what you see, the bad genotypes, before we refine them, there were a lot more things that shouldn't really have been there in terms of non-synonymous mutations that caused differences in the protein. There are a lot more of those than you'd expect to find. But once you do genotype refinement, the whole plot shifts to what you would expect to see. That most of the mutations that you find in exons are actually synonymous, and the ones that went away all were the ones that we'd expect that we did not expect to be there in the first place. Okay, so we kind of skew now towards normalcy after we refine the genotypes, and we see the kind of the distribution that we would expect to see. All right, so that for now is this part of the process. We have filtered genotypes, we've refined them, and finally we have a call set that we can go sit down and tell you whether it's a good call set or not. And with that, I'm going to take questions and ask Aaron to come in from the other. Oh, and here's Aaron. So questions for anyone about genotype refinement? Great. So Aaron will talk for about 15, 20 minutes, then we'll break for lunch. So he's the only thing standing between you and lunch. Okay, so Anton is saying when Aaron is done, do not leave. Anton is going to have you start up your environment so that when you come back from lunch, it will be ready for you for the hands-on, because he's going to start immediately after lunch. All right.
Good. All right. Well, I apologize in advance for being the one person between you and lunch. Two people. You're right. It's not just me. Thank you, Anton, for sharing the blame. Okay. So at this point, we've pretty much gone through all the best practices, right? We've gone through, we've got our raw reads, we've mapped them, we've taken care of our duplicates. We actually went in, did the haplotype caller. Maybe we went in as well, did some joint calling, filtered our models, and we're done, right? All go home. So the thing is, let's assume for a second that everyone right now listening is a professional. We trust you all. You've done everything methodologically correctly. You haven't let your cat walk across your keyboard. No grad students have lost samples. Everything is fine. That still doesn't mean, of course, that you've actually correctly identified every single variant in there. Like, ultimately, you have a certain amount of calls, but in a perfect ideal world, right, all of our calls would align with biological truth. And every single thing that we called as a variant would only be true. There wouldn't be any particular mutations, no noise in the data that happened to look close enough to a real variant that we didn't catch them. But that's not the case. The truth is, you don't want to think in absolutes. You're going to fall somewhere between terrible, everything you called with an artifact, go home, or perfect. You found every single thing, every call was perfect and correspond to underlying biology. The question is, how can you tell where you are? Right? Like, there's no source of truth. That's why you're doing the sequencing, is because you're trying to determine truth. How do you determine how good your call set actually is? And well, the answer is, for the most part, there are a certain classes of group statistics that you can compute on your final call set. We'll go over each of those in detail: the number of SNPs, number of indels, the TI-TB ratio, indel ratio, and concordance. I'll cover every one of these. And the important thing to gather, though, you're going to try to find another call set to compare to. And finding the correct truth set is actually pretty important, because the statistics that you compute here, they won't line up. There are lots of good call sets that will have different statistics than yours. So what we're going to go over a bit is how to find the correct truth set to compare to, because it's really important. Like I said, we have no source of truth in advance when you sequence someone. That's the purpose of sequencing, is to try to find that. So to compare, you have to find something that's going to provide a good equal comparison. So as I was mentioning, one reason why it's important to match them really well is because the statistics change depending on several parameters. One is just, well, population ethnicity. It actually matters. We'll go into that in more detail later, but like, things like the rates of actual SNPs you see in a person is not constant. Now, it's somewhat constant per population, right? As we mentioned, there are relatively few true de novo mutations for each of us. Most of our variants we get from our parents, which means that between generations it's reasonably stable. But so within subpopulations, it tends to be stable. But across the entire human race, I mean, the number of mutations I have is likely different than Anton's or Eric's. It's just the nature of things. Also, the statistics you see depend on exactly what kind of sequencing you did. I mean, you're just doing like exome sequencing, that's going to look different than whole genome. Partly, you see different distributions and depth around there. You see different error rates around the edge of those distributions. It's going to look different. Not to mention that when you're only looking at specifically protein encoding regions, the amount of mutations in those regions can be different than looking at other parts of your genome. There's, some regions are more conserved than others, as you well understand. Also, again, when talking statistics, you should probably try to keep the cohort size reasonable. Certain statistics shift a bit if you're dealing with, calling 10 people versus 10,000. Now, in general, of course, you have to find where will you look for your truth sets. We mentioned a couple of these earlier. I won't go into great detail on them. Eric mentioned dbSNP is there. It's there. Sometimes you can do a quick check against it. The truth is, as individual PI submitted data, there's not that much quality control going in, so you can't use it as a 100% source of truth. GATK and Nomad are pretty solid. Like, those are pretty well curated. There's a lot of work to get them to make sure we're confident in those. GATK, that's actually just exome data, though, so not whole genome. As we mentioned earlier when Emma was talking, Nomad has a lot of whole genomes in it, though. That's a really good one to compare against. HapMap is solid. HapMap, Omni, and Genomes in a Bottle are good because they're also public databases. They're really solid, but very well curated. And specifically mentioning GATK a little bit, I think the Exome Aggregation Consortium, I think is what it stands for, they actually took together a lot of different independent projects and they broke them down by ancestry. Someone asked me yesterday in joint calling that how you can tell your data is fine, I guess, when you potentially mix different people together. And I think I mentioned to that person that you can do analysis later. You can separate by likely ancestry and compute statistics based upon that. And this is an example. In GATK, they broke down these other projects by ethnic group, and you can see that, for instance, the number of SNPs really vary. I likely be up here at the top in the red area. Like, you see that in African populations a lot more. But even along the baseline here, you see some significant differences there. Like, I mean, it might not be as dramatic, but you can see your baselines. They're different. Okay, so identifying a good one to compare against will give you a lot more confidence. And why do you see them? Well, of course, there are biological reasons, right? Older populations have a tendency to have time to build up more and more, variants there. You also have founder effects, when a small population leaves, you got that some, tightening of the genome and then recompression there. There are a bunch of things that happen. There are reasons why, for instance, we see these patterns, but we need to be aware of them. So what are the metrics again? I'll go over all of them in detail in the coming slides, but you have roughly the total number of indels and SNPs, the indel ratio, with a cool one called the TITV ratio, and just genotype concordance. Now, as we mentioned, it's useful for a basic sanity check. What if you're comparing like populations? You want to look at roughly the number of total indels and SNPs you're looking at, because we know this for the most part. For given populations, we've done enough large call sets to have a reasonable baseline. Now, I should note as well, when you're comparing populations, sometimes you won't find a perfect match. You just won't find it. But what we often do is we take subsets of our call sets and might compare against subsets of another call set. That's actually completely valid. All right, often we'll use a subset of our call set just as a proxy for the entire thing. I'll talk a little later at concordance. This is what my team does when we're doing our last stage of calling for ALU, for instance. They're literally generating those statistics. The team messaged me this morning, some of the initial ones, okay? You compare a subset with others, you can get good initial numbers, all right? So assuming you can match a subset, a good metric is just looking purely at the number of indels and SNPs. If it's off by like an order of magnitude or something, or you're expecting 4.5 million and you see like 100,000, okay, something's probably wrong. Another one is the indel ratio. Now, this is cool. A lot of these rely on us understanding a little bit of what's happening in the underlying biology. So in a human, insertions and deletions don't actually happen at the same rate. You're actually much more likely to have a deletion than an insertion. Now, over time, in areas for really common mutations, they tend to balance out. In really old variants, you tend to have about roughly an evening out of insertion versus deletion ratio. But in novel and rare mutations, you still see a heavy bias toward deletions. Right? They haven't had time to be acted on by natural selection. When you randomly delete things, as it turns out, that's often bad. Whether it be in your genome or somewhere else. So you can actually look at that ratio. You find a population to compare against, and you look at in your call set, well, what was my indel ratio? What were my insertions versus deletions? And if it's and you can also break it down by your common variants versus your rare ones. And if you see numbers that look correct, it's another bit of evidence. Okay. And I should point out that all of these are just adding confidence to the fact that you have a good call set. I mentioned 4 specific ones here. These are like the minimum in a real way. If you have better metrics, for your particular case, I really do suggest you use them. But we also suggest at the very least, you can do something like these 4. All right? Another one is the TITV ratio. Again, this is cool because it's another situation where we know something about the underlying biology and we can use it to try to see if our data matches up with it. So I'm a computer scientist. That's my thing. I love abstractions. If I can, I don't know. Before that, my background was physics. There are jokes about us treating cows as perfectly frictionless spheres and stuff like that. A good abstraction helps you so much, okay? But in this case, when we talk about the genome and nucleotides as C, A, G, and T, we're actually ignoring something. There is underlying chemistry at play here, okay? We kind of divide our nucleotide into 2 classes. You have the big ones that are pretty chemically similar, A and G, and you have the smaller ones that are chemically similar to one another, C and T. If you look at all the possible ways SNPs can occur, in nature, going between the chemically similar ones happens 6 times more likely than going between the chemically different ones. Now, the truth is, if there is just a glitch somewhere in processing, it doesn't know that. Like if you're dealing with letters in the screen, there are errors in sequencing. It won't respect the fact that there is that bias in nature, especially because if you look at the total number of possible transitions here, 2 of them, like from A, A to G or G to A, that's only 2 possible transitions versus from A to C to T and back and forth. There are twice as many transversions as transitions. Assuming just noise in the data, you're going to see transversions occur twice as much. But again, in nature, you see the opposite. Transitions occur. So you can look at that ratio. You look at that, you can calculate that ratio in your call set, and you can say, does my call set match what I expect to see in nature? Or does my call set look like I'm seeing a lot of noise? It's one of those cool metrics where, has been a theme in many cases, we pull in additional information to better give us context to interpret what we're seeing. And important down there, again, in humans, a lot of what we say is in humans, you can look at, it differs slightly. We mentioned grabbing the right truth set, right? It differs between looking at the whole genome versus just exomes. You see slightly different numbers there. And the last one is really just genotype concordance. Now, we mentioned, for instance, comparing call sets to one another. If you have a truth set, seeing how well you match up with it. These are the numbers I mentioned. I just got back just this morning, for instance, from the next call set. What you often do is, I said we often don't have a source of truth, and that's true. But what you can do, like Eric mentioned earlier, is take a sample where you have a source of truth, like good old NA12878 or a chimeric sample or something else, and we had about 8, and you run them with your call set. You carry them through its controls the whole way through, and at the end, you extract that subset, and you do a direct comparison to a source of truth. And you say, okay, how well did my handling of these known samples here compare to what I independently know they should be? And you can calculate a few statistics on that called concordance. So for instance, I'll go into a little more detail later about exactly what some of those statistics are. It's worth noting that you won't always have a source of truth like that. Maybe you're not doing human sequencing, right? One of the things you can do to simulate a truth set is actually run the samples as much as possible through different sequencing technologies, similar ones, right? Like you don't want to jump too much, but even among short read technologies, different ones have different characteristics. Sanger is good; it's expensive but really high accuracy. Different technologies have their own different sources of biases. If you want to tell whether a particular read and processing was good, you don't want to run it through the same exact pipeline twice. You just reinforce your biases. Ideally, you want to run it through two separate pipelines and then compare those to each other. Is one way of pretending you have a truth source, giving you another independent source of information to check your call set against. Okay? Now, speaking of concordance, it's not particularly hard. What concordance does, it treats one sample as truth. Let's say you have a series of, the numbers are made up, of course, in any real sample, the numbers are huge. But let's say you have a source you're treating as truth. Let's say it's a sequence for, I don't know, maybe anyone to 878. And you have a certain number of variants you know are in there, and you run anyone and 1 to 870 through your own sample as well. And you have a little Venn diagram. You can see where your numbers overlap. You can see that, okay, you treat the overlap as true positives. You called those correctly. But then maybe there are some that you should have called and you didn't. Maybe the filtering model yanked them out. Maybe you just didn't get good reads on them. Well, okay, those are false negatives. And maybe there are novel variants that you're calling inside of NA12878. And really, again, she's been sequenced so much, we're pretty sure that's probably an error. So we're treating those as false positives. And you can just calculate, for instance, on your sensitivity, what fraction of the variants that should be there did you get? In terms of false discovery again, what fraction of the variants what fraction of the variants in your call set were actually present in the original one and should have been there? So you have to want to look at your false positives over again, the false positives plus the true positives. Basically, which ones are false versus all the variants you called. These are good metrics to see basically how far off were you. And finally, it's not purely about the variants there. You want to look at genotypes too, because seeing a variant is one thing. He mentioned there's a difference between just site quality versus genotype quality. Sometimes it should be HOM, sometimes you call them het. We also have a genotype level concordance we can run too. This is what we are running, for instance, just this morning. We're running the numbers against a couple of control samples, and you get good numbers out of it. You want to see high concordance with known truth sets. If so, you have a lot of confidence that you've done something good in your call set. All right. Because you have the slides, I don't have to go too much into the actual commands here. We have the commands. There are tools, tools in Picard or inside of GATK, and we actually talk about why you might want to use one versus the other. Okay, sure. But in the interest of lunch, I don't have to read everything on these slides. You have them yourselves. But again, those are basically 4 types of statistics that you can run. And when you're done with it, ideally, you look at your numbers and say, well, okay, if you've chosen your truth set right, you say, I know everything isn't 100% okay, but I have confidence that nothing egregious happened. And you can go from there. I hope I managed to come in under time. I really hope I did. I'm trying my best. So any questions? Everyone's so ready for lunch. All right, and before we go though,